We're getting into the real heart of genetics now. We're going to start historically at the beginning and then continue through with our story as we learn more and more about genetics. So to start it all out, Mendelian genetics. Back in the 1800s, there was a really popular theory of inheritance called the blending hypothesis. And this basically said that when two parents mixed and had a child, the offspring were a perfect blend of the parents. So if you had a blonde and a brown-haired parent, they would produce a dirty blonde child. There's a lot of reasons why this is flawed that don't have to do with the actual science. One of them being, if that was the case, then over time you would see all other types of traits disappearing except for that blended trait. Also, it doesn't really fit because we've seen that sometimes a trait won't show up for generations and then all of a sudden it'll reappear. So our real understanding of genetics began with Gregor Mendel. He was an Austrian monk in the mid-1800s, and he is the reason why we understand genetics as we do today. He was from a gardening family, a farming family, and he really wanted to be a teacher, but he couldn't afford to go to university, so he went and became a monk. And in the monastery, he was able to tend to their garden there and kind of explore his own ideas and theories and do his own experiments. So he had some really great successes. He was very meticulous in his experimentation. He followed almost essentially traditional experimental methods. He kept detailed records and did impressive quantitative analysis of those records. So he really furthered our understanding of the field before it really even was a field. And this is Gregor Mendel right there in that picture holding a little plant. Thought that was pretty cool. Now, what was his experiment? He was cross-pollinating pea plants. What he would do is remove the stamens from a plant. These are the pollen-producing parts. And that way he could capture the pollen and very specifically choose what plants to breed. They weren't breeding on their own. He would then take a little brush, get some of that pollen on it, and then transfer that pollen to the stigma, the part of the plant that can be pollinated and fertilized. He then let the plants grow and produce seeds. He collected all the seeds and planted them, and then watched as the offspring grew up to see what kind of traits they produced. So why peas? Well, he picked peas for one major reason. Their physical differences are very stark, and they tend to only have one of two options for their traits. They can be tall or short. They can have round or wrinkled peas, yellow or green peas. So this was a really great way for him to be able to see differences right away. Each variant was a trait, so he collected data from 7 different traits. And he used something called true breeding individuals to start all of his experiments. True breeding individuals are just those whose previous generations were all the same type of plant as them. They all had the same trait. So if you had a tall plant, all of their parents were tall, and all of their parents were tall, and all of his siblings or its siblings were tall. So he took 2 true breeding plants with opposite traits. The example that we're going to follow is this color of the flower. So a purple flower, and bred it with a true breeding white flower, meaning its parents were both white flowers. And the purple parent were both purple as well. So we called this generation the P generation, or the parental generation. The offspring that they produced were the F1, and then when those were allowed to self-fertilize, they created the F2 generation. And the F stands for the Latin term filial, which just means sons or children. So this is the first children generation, the second children generation. So the F1 generation, the first generation of offspring that's produced, they're called hybrids. They're the mix of the two traded parents. So when we look at this particular example, the purple flower being crossed with the white flower, the F1 generation produced all purple flowered plants. And he noticed this for no matter what trait he tested. There was always one trait that seemed to overpower the other. The purple flower overpowered the white flower every single time. There was never a white flower in that generation. He then allowed the F1s to self-fertilize, so they would mix their own pollen and their own eggs together to create offspring, and created that F2 generation. And what he saw was that 3/4, or about 75%, were purple, but then 25% produced white flowers. So that hidden white flower trait was able to pop back up. He had a 3-to-1 ratio. So what was going on with all of this? Well, nowadays, those different variations of the traits, we call them alleles. You have an allele for brown hair, an allele for blonde hair, for red hair. They're just different variations of the trait. And these genes are found at a specific locus or location on a chromosome. So in the example here, you can see that the locus is the same on both of the homologous chromosomes. The gene for flower color is found in the same place. However, this one has an allele for purple flowers, while the bottom chromosome has an allele for white flowers. What is causing the difference between those two? Well, what it is a difference in the nucleotide sequence. The actual order of the DNA bases is going to be different in each of those. You might have one that has an ATCG, and the next one's going to have ATTG, something like that. So they just have different letters to create that same gene. There's a mutation in one of them, essentially. Now, diploid individuals, like humans, we inherit a chromosome from each parent. So you get one from your mother, one from your father, and those come together to create you. You inherit both copies of those chromosomes. That's why we have 23 pairs of chromosomes. Now, with Mendel's results, he showed that there was always one trait that seemed to overpower or mask the other trait. And when we look at all of his different traits that he was following, we can see in the F1 which ones were the dominant trait. The dominant allele was the one that overpowers the recessive allele, the hidden allele. These dominant alleles are considered to be the wild type ones. These are normally functioning proteins. The recessive alleles are the ones that are hidden, like the white flowers, like the green and wrinkled seeds, the dwarf plants, things like that. The recessive alleles, you can only see a white flower if you have both copies of that recessive allele. You get a recessive allele from your mother and from your father. These ones are not considered wild type. They're considered mutant alleles. Another way to say that is just they're alternatively functioning or non-functioning proteins. The protein isn't working the way it was originally designed to. It's a different version of that gene. So we use these terms today, genotype and phenotype, to talk about what Mendel found. The physical appearance of the trait is the phenotype. PH for physical, PH for phenotype. And then the genotype describes the genetic makeup of that organism. What alleles did that organism inherit from its parents? So the genotype is represented by 2 letters. And it's just whatever letters you inherited from your parent. The dominant alleles are represented by capital letters, and the recessive alleles are represented by lowercase letters. So true breeding purple plants would have the genotype big P, and the true breeding white plants have the genotype little p. So now what would that make the genotype of the F1 generation, that filial 1 generation, and how would we know? So the way we do that is using a tool called a Punnett square. And this just allows us to look at the possible outcomes of crossing two individuals. We can see what's the probability of having an offspring with this particular trait. We can even use it to determine the ratio of our genotypes and our phenotypes from that particular trait. When you're doing a Punnett square, one side is going to represent the sperm. So what possible alleles are in the sperm? And the other side is going to represent the egg. What possible allele is in the egg that's going to be mixed? And with these, remember that your genome, even if you are a diploid individual, is going to be halved during meiosis. So in meiosis, those alleles get separated. So we have to look at one allele at a time mixing with the egg and the sperm. So with genotypes, you can have a homozygous genotype. Homo just means the same, so you're looking at an organism where they inherit two of the same allele. You will have homozygous dominant, where you have both copies of the dominant allele, and you can have homozygous recessive, where you have two copies of the recessive allele. And then when they're mixed, it's heterozygous, that word hetero meaning different. So this is the case of the middle plant here. It has a capital P and a lowercase p. So in this case, we're inheriting two different alleles from the parent. Now, how can you tell if an organism that shows the dominant phenotype, are they homozygous dominant or are they heterozygous? How can we tell just by looking at the plant or the organism itself? Well, you can't. You'd have to do what's called a test cross. You're going to cross that individual of unknown genotype with something that has the recessive genotype, where we know 100% what its genotype is. And in particular, you want to use the recessive one. That way you can see if the combination of that little recessive allele with both parents creates that recessive phenotype. So if this was a homozygous dominant cross, then in every single case, you would have a dominant allele being given to the offspring. So it would hide the recessive allele from the white flower every single time, and you'd create all purple offspring. If we had an instance where there was one recessive allele that could have been given to the offspring, then you have a case where half of your offspring are going to show that recessive allele. That would conclusively tell you that you were looking at a heterozygous individual. Now this shows us one of Mendel's laws of heredity, the law of segregation. He says that during meiosis, the alleles are going to segregate, another word for separate from each other. If you have two dominant alleles, you're going to put one dominant allele in one of your gametes and the other dominant allele in the other gamete, and so on and so forth, so that each allele ends up in its own gamete. And this is going to happen between metaphase 1 and anaphase 1 when those homologous chromosomes are separated from each other. Now, this was seen when Mendel was doing what are called monohybrid crosses, where he was looking at one trait specifically. However, he extended some of his work to do experiments on two traits being inherited simultaneously, such as looking at peas that were both yellow or green and round or wrinkled. He looked at two traits at the same time. So how did the results for these ones differ from his monohybrid crosses? He took true breeding green wrinkled peas and crossed them with true breeding round yellow peas. Essentially, they had genotypes that were all dominant or all recessive. And that created a mix of big Y, big R, little r, the heterozygous mix in the F1 generation. You can see that in our cat example here. They have a mix of all of them. Now, what he found was that there was 9 yellow round peas, 3 green round peas, 3 yellow wrinkled peas and 1 green wrinkled pea in the F2 generation when he allowed the F1s to self-fertilize. So we were crossing 2 heterozygous individuals with each other. How did he get that particular ratio? We're going to look at 2 possibilities here now. So the first thing that we need to look at is, how are our gametes formed? If I have 2 heterozygous individuals, how do I create their gametes? Do we have a situation where I'm looking at one gamete with big R and big Y, and the other gamete with a little r, little y? What's going on here? So we're going to look at this example first and see if this is possible. When we do a Punnett square and we cross those gametes, we get 3 round yellow peas and 1 green wrinkled pea. Well, that particular ratio does not match the one that he saw in his plants. So this cannot be the way that the gametes are created. So with these traits, the genes are on different chromosomes. They're not connected in any way at all. So that means when they make their gametes, nothing is linked together. So what we end up having to do is we have to foil our particular organism's genotype to get the possible mix of gametes, just like you do in math. So if I have this particular individual here, the way I foil their genotype to create the gametes is just like you would do in math. The first, which would create big Y, big R, outer, big Y, little r, inner, little y, big R, and last. So you would have little y, little r. And those are your possible gametes for one of the organisms, the father or the mother. You would have to do it again for the second organism and figure out their possible gametes. Well, in this case, we were crossing 2 heterozygote individuals, so their particular gametes are going to be identical. And now, when we do this particular cross, we start to see that 9 to 3 to 3 to 1 ratio that Mendel saw in his pea plants. You have 1, 2, 3, 4, 5, 6, 7, 8, 9 plants, or 9 peas that produced round, smooth yellow peas. You had 3 that produced yellow wrinkled peas, 3 that produced green round seeds, and then the 1 that produced the green wrinkled seed. So this is called a dihybrid cross, where we're crossing 2 different hybrid traits at the same time. So this now leads us to Mendel's second law of inheritance, the law of independent assortment. He was able to figure out that every single trait, every single gene is going to assort independently, meaning every single chromosome is going to go into its own egg. We're not going to have different traits on different chromosomes that are matched up that follow each other. Everything is separated randomly. Now, we do have a situation where that's not true, and that's when we talk about genes that are on the same exact chromosome, like this one over here. When I have 2 genes right next to each other on the same chromosome, more often than not, they are going to be linked. This is because they aren't going to be separated from each other. Every chromosome moves as its own unit. Now, the only way that they could ever possibly separate from each other is during crossing over, during meiosis, when those homologous chromosomes reach over and switch genetic information. So you could potentially have a case where the genes do get separated when they're linked closely to each other. So these 2 are so close that they're probably never going to get separated. However, these ones are not linked. They're so far apart that there is a chance that crossing over will occur somewhere in between the 2 of them. So they're not considered linked genes. They're going to assort independently, just like Mendel said. These linked genes are not essentially common, but they're just a way that Mendel's laws were thrown for a loop. Now, normally, Mendel's law of independent assortment says that every gene is going to assort independently from the other. It's going to move into its own gamete no matter what the other genes around it are doing. Now, Mendel was actually very lucky with his choice of pea plants. It might have been that he picked peas because he was from a farming family, and peas are legumes. They will end up helping with nitrogen fixation. They'll put nitrogen back into the soil so it keeps your soil healthier for longer. Maybe he just really liked peas, but scientifically it was a very lucky choice. Most plants have many different possible traits per gene. So you won't just have a purple and a white flower. You can have many different color flowers, many different shapes of flowers. So peas have what we would say are relatively simplistic genes compared to most plants. He also was lucky in that all of the traits that he picked were single genes. None of them were linked. None of them had any other types of inheritance patterns. It was strictly dominant or recessive. Most organisms are not that simplistic, and that's going to be the focus of our next lecture. Now, the last thing we're going to talk about today are pedigrees. I absolutely love these. They're really fun. I feel like a detective when I'm trying to read a pedigree. All they are essentially tools that are used to track a specific trait through a family. And they look like a family tree because it's pretty much what they are. You can use it to track a trait and see if it's a dominant one or a recessive one. In later lectures, we're going to look at, are they X-linked or are they on a normal chromosome? So when we're looking at a pedigree, every single line represents a generation. And usually they're going to be numbered individuals within those generations. So that way I can ask you, hey, what's the genotype of the person in generation 2? Number 4, and then you know exactly who you're looking for. Go to generation 2 and find number 4. So when we're looking at these pedigrees, they're laid out in a way that's really easy to understand. Squares represent males, circles represent females. Lines connecting males and females that are horizontal indicate that this is a mated pair, or in the case of humans, you're looking at a marriage. When you have offspring, there's a line vertically that comes down from that mated pair and then connects all of the offspring together, just like this here. So they had first a daughter, then they had a son, then another daughter, then another son. These are the spouses of the children that generation 1 had. So they're not necessarily part of generation 1's family, but we include them because in generation 3 we're looking at the offspring of generation 2 and we need to know what their parents were. When you have a shaded in circle or square, that person is going to be affected. They're going to have the disease or the trait or whatever it happens to be that you are tracking. So as I said, we use these pedigrees to figure out whether the trait that you're looking at is a dominant or a recessively linked allele. Now, with dominant traits, the key thing is it does not skip generations. You are not going to see a child that has it and their parents are unaffected. If a child has it, they're going to have at least one affected parent, potentially 2. Because we're talking about a trait that's linked to the capital letter, the dominant allele. So you can have a homozygous dominant individual or a heterozygous individual showing the trait. So in order for you to have the trait, you have to have one of those capital letters. That means you have to have gotten one of them from your parent. If your parent has a capital letter, they have the trait as well. Now, an example is achondroplasia. This is a form of dwarfism. So it's actually a dominant trait. That goes to show that just because it's a dominant trait does not mean it's going to be the most prevalent one. You can still have these traits that don't show up very frequently in the population. Another example is Huntington's disease. This is a degenerative brain disorder. So somewhere between your 30s and 50s, you will begin to show symptoms, and your brain essentially degenerates to the point where you start forgetting how to do things, and you slowly lose control over your thoughts, over your body control, and eventually it is fatal. So when we're looking at the pedigree for traits like these, you will notice that every single child will have at least one affected parent. That's the key thing for these pedigrees. Now, one thing we can do with pedigrees is not only determine is it dominant or recessive, but you may be asked to determine the genotypes of the individuals inside that particular pedigree. The first thing you have to do is determine how it's inherited, because you don't know if the shaded in individuals are carrying the dominant allele or the recessive allele until you figure that out. So find out what is the inherited individual, or how is the trait inherited first. Now, for this particular types of traits, the next thing I want to do is determine who is either the dark circles or the free circles, because if it's a dominant trait, your free circles are going to be the homozygous recessive. If it's a recessive trait, then your affected individuals are going to be the homozygous recessive. So in this case, we're looking at a dominant trait, so all of the empty circles and squares are going to be homozygous recessive. So I can go in and automatically fill in all of those individuals there. And just from here on out, all I have to determine is, are my individuals going to be homozygous dominant or heterozygous? That's what I have to figure out now. I know that every single one of these people has to have at least one dominant allele because they carry the trait. So the way that you can figure out whether or not they have the lowercase allele is by looking at their relatives. Number 9 is going to be the simplest to figure out first. That's because they have to get one allele from parent number 3. Parent number 3 only has little a's to give, so that means number 9 automatically is going to be heterozygous. When we're looking at number 8, the same thing applies. They received 2 little alleles. They are homozygous recessive. They only received one of those from their first parent, so the second one had to come from the other parent. So number 4 has to be heterozygous as well. Now the same thing is going to happen for the case of number 7 and then parents 1 and 2. Number 7 here had to receive each of these alleles from their parents. They couldn't just appear out of nowhere. So both of the parents are going to be heterozygous. But now we run into a weird situation here. When I'm crossing 2 heterozygote parents in a Punnett square, I end up with a lot of different possibilities. I can have homozygous dominant children or heterozygous children, and I don't know just by looking at these particular people if they carry the recessive trait or not. It would help a lot if they had offspring for us to look at and we could see, do any of their offspring have the recessive alleles or not, but we don't have that luxury now. So really, we can't tell these ones conclusively, and that's an okay answer. You would just have to say that these individuals could be homozygous dominant or heterozygous. Now, recessive traits, the shaded in individual is going to be linked to the recessive allele in this case. So everybody who is shaded in is going to have the 2 little alleles. And in these ones, because it's linked to the recessive allele, we are able to skip generations. And that's because there are going to be individuals that are carriers. The heterozygous individuals are called carriers. They will be able to pass the disease on. They have the ability to give a child that recessive allele, but they themselves will not show any of those recessive traits. They're going to show the dominant trait because they carry one dominant allele. So two examples of traits that are linked to recessive alleles are albinism. This is where you cannot produce melanin, so your skin is very pale, essentially white skin. You'll have white hair and pink or blue eyes. And then there's also cystic fibrosis. This is where if you inherit both of the small alleles, you're going to have poorly functioning or non-functioning chloride ion channels. And we've talked about this a little bit when we were talking about cell signaling. Basically, if you can't pump chlorine in and out of your cells efficiently, you're not going to osmoregulate properly. You won't be able to control the water that's in or around your cells. And as a result, it's going to produce a really thick mucus around our intestines and inside our lungs. And these particular individuals will have such thick mucus that it's going to affect normal functioning of those organs. They won't be able to digest food properly. They won't be able to breathe properly. So cystic fibrosis is considered a fatal trait. Now, when we're looking at a pedigree and we're trying to figure out what the genotypes are, remember first you have to figure out, is this a recessive or a dominant pedigree? If you are not told that, you're going to have to look at how the pattern is inherited. In this case, if you look at number 17, they have two unaffected parents, but they are affected. So automatically this cannot be a dominant pedigree, because if it was dominant, they would have to have one affected parent. And you can see that number 8's parents, 1 and 2, one of them is affected. So this skipped a generation. That's characteristic of a recessive pedigree. So now the next thing that I do is figure out, okay, who has the trait? Who can I easily label with either 2 little a's or having at least one dominant allele? Well, in this case, the shading in is tied to the recessive trait. So I can shade in every single filled in person with 2 small alleles. And every single person that's not shaded in has at least one dominant allele. So it's just the opposite in coloration of the last pedigree chart. Everything else is going to be fairly similar. You have to look at the parents, you have to look at the offspring to be able to figure it out. So starting with number 13 here, number 13 has 2 small alleles. They have one parent with 2 small alleles, but that means number 6 has to have a small allele to be able to give to that child. Now this child has to have, number 14, has to have a small allele as well, because that's all that number 5 can give. When we're looking at this middle family right here, number 17 has the disorder. They had to get one of those alleles from each of their parents. So number 8 and number 9 have to be heterozygous. Now the offspring, we won't be able to tell, because when we do that cross again of 2 heterozygote individuals, we're not 100% sure which one it could be just by looking at them without knowing their offspring. They could be homozygous dominant or heterozygous. For number 7 here, they have a parent who is homozygous recessive. That parent has to give one of those alleles to the child. Therefore, number 7 has to be heterozygous. And when we're looking at parent number 2 of this family, just to round out that side of the pedigree, every single child has a small allele, but every single child also has a dominant allele. So most likely, this parent is homozygous dominant. However, We're not 100% positive because you have to do a Punnett square for every single child. So there is a high likelihood that they are going to have a kid with the trait showing up if the parent is heterozygous. However, it's not impossible for them to have kids who don't show the trait at all. So in this case, it's probably safer to say that you can't tell which is which, but it's almost positive that you're going to be homozygous dominant. Now, the very last part, the right side of this family pedigree, individual number 4 can only give a little allele, so that means number 12 has to be heterozygous. And because number 10 is homozygous recessive, that means number 3 had to give a little allele, so they have to be heterozygous as well. And that's all I have for you guys. If you have any questions, just let me know.